Hello everybody, welcome to a video on COVID-19. We're going to treat this like a clinical application, so there will be no test yourself. The next module is the true clinical application for the lymphatic system and immune function. But if we're going to pick one subject in this entire world, the entire planet Earth, that we'd have an extra clinical application type module for, it would presumably be COVID-19. A disease, a viral disease caused by the SARS-CoV-2 virus. That's just a type of virus. SARS standing for sudden acute respiratory syndrome. If you were around in 2002 and you were paying attention or able to pay attention, meaning you weren't an infant or in your infancy, then you would have known the SARS outbreak of 2002 was a pretty big deal, and it's acute respiratory distress caused by a virus. And we'll talk about the mechanisms in just a bit. Structurally, CoV-2 is not a complex virus. Most viruses aren't. There's about 15, maybe something total proteins. We're really going to worry about 7 main parts, and I'll just talk about each one because each one of these is fairly functional. So the spike protein and the hemagglutinin esterase protein in red and blue are these proteins sticking off of the viral capsule. M protein and the E protein, and then in dark blue, a little hard to see against the black, but I tried to make in dark blue. There's an outer casing here. There's an outer dark blue casing, that's the envelope, and then in bright green inside is the nucleocapsid proteins. They're like an inner capsule. And then there's a single-stranded RNA. It's a positive sense, but single-stranded RNA. Nucleotides come, of course, in DNA and RNA. And RNA is single-stranded in this case. And that is the genetic material of the virus. So the spike protein and the hemagglutinin esterase protein are really the two most important, because those are the two that allow the viral particle, the virus, to grab onto, to attach or adhere to the target cell. And not only that, but to drill in and spiral into the target cell to allow the virus to enter the target cell. And that is viral infection. So the spike protein and the hemagglutinin esterase protein act like little, I don't know how you want to think of, grappling hooks maybe, that allow it to grab on, and then little drills to drill through the cell membrane and get inside of the cell, and everything comes from there. Okay? And what do they bind to? What are the host cells? Well, ACE2, right? And that's going to be angiotensin converting enzyme number 2. We've seen this enzyme briefly, but in no detail, so you don't need to sweat it. But when we talked about the RAAS, the renin-angiotensin-aldosterone system, when you were trying to combat low blood volume, remember back in 40B, and we were talking about how angiotensin 1 is converted to angiotensin 2. It's actually done in the lungs, believe it or not. And so it's the ACE2 cell receptor on a very special type of lung cell. It's going to be the type 2 septal cell. We saw that back in 40B too. That's where these viruses are going to be most happy attaching to. There are ACE2 receptors on tons of body cells, which is really great, but not so much great. I'll take my little smiley face here and maybe kind of cross it out, because the more ACE2 receptors there are, the more places that this virus can bind to and adhere to and penetrate and enter more host cells. The more ACE2 receptors there are, the more viral infection you're going to get. And the type 2 septal cells in the alveoli are a huge one. So that's spike protein and hemagglutinin esterase protein for kind of binding. And again, we talked about antibodies. The spike protein is a colossal antigen of interest in this virus. What if you could build an antibody that could come along and it could bind to the spike protein or the hemagglutinin esterase? But I'll show it on the spike protein. Well, do you remember neutralization? That protein is essential for boring into and entering and adhering to the host cell. If you can cap that thing, you've just neutralized the virus. So needless to say, that spike protein is a very, very popular antigenic target for antibodies. The M and the E are a little less fancy, but still really, really important for the viral health and the viral survival and the viral propagation. Because once the virus enters the cell, it sort of breaks into its constituent parts, and then it runs the RNA through your host ribosomes. So the RNA goes through your host ribosomes, and it makes all the viral proteins. It makes more spike protein. It makes more hemagglutinin esterase protein. It makes more nucleocapsid proteins. But those are kind of sitting around floating in space. It's the M and the E protein that go to work and they reassemble the virus in multiple copies inside of your cell. So you're reassembling the virus inside your host cell, and then from inside it destroys the cell and so on. And then it goes on to infect the next cell and so on and so on. And that is viral transmission. And of course, you're very familiar with this. If the virus is in your upper respiratory tract, you are going into the epithelial cells, the mucosal epithelial cells of the upper respiratory tract, and you're blowing those cells apart. And you're just blowing them into liquid mucus. And of course, that becomes a runny nose and cough and so on and so forth. We know factually that the virus emerged in China in 2019, and we know it's become a global worldwide panic. But as scientists, our job is to help and to cure and to learn and to study and to understand. We don't sit around pointing blame about who did what. That's just for a whole other class of people that are not us at all. So we just worry about the origins. That kind of helps us with the zoonotics. I'll talk about that in a little bit. Animal transmission, so on and so forth, or hybridization from an animal host, so on and so forth. The only possible alternatives or competitors to this would be in modern recorded history, the smallpox epidemic, which typically gets a date about 1877 to 1977, about a 100-year period in which it's reportedly killed nearly 500 million people worldwide. The AIDS epidemic starting in the very late 1970s, the early 1980s, and then of course the closest analog was the Spanish influenza, which lasted about a year and a half, also about 1919 to 1920, and wiped out massive fractions of populations because it was incredibly virulent, even more virulent than COVID-19. And that was just about 100 years ago. And there are 4 genetic strains so far. And again, there's mutation from strain to strain. The virus evolves from strain to strain. We talk about evolution being 1,000 generations. So evolution taking course in 1,000 human generations. Call the average human generation 25 years. So 25,000 years for some trait to really fully show distinct evolution across the human span. In viruses, that's a matter of months. 1,000 generations is a matter of months. And so you can get significant evolution. And starting with the alpha, and then the beta, and then the gamma, and then the delta. And the beta coronavirus is the one that was essentially responsible for the second wave of 2020. And hello, delta virus, delta corona. That's where we're at right now, right? And so in late 2021, we're at the delta, and hopefully we will not go to the epsilon or the zeta and so on and so forth. But as it evolves to strains, and they're named along the Greek alphabet. So that's why we're talking about the delta variant at this current point, summer 2021. And the zoonotics is always about an animal host. Bats, all sorts, primates, all sorts of other mammals are great hosts for these viruses. And then the question is, can it jump? Or pigs, or even birds, avian flu, swine flu. They have names like that for a reason. They are in zoonotic. Zoonotic just means from an animal host. Zoology, a zoo with animals. Zoo is a root for animals. So zoonotic means from an animal host. And the bat has a beta coronavirus called RATG13, for whatever that's worth. It's 96+% similar genetically to the human beta coronavirus, the COVID-2 that pushed that first surge of COVID-19 in the year 2020. So possibly was it hybridized from the bat, so sort of the virus inside the bat sort of hybridized and became a human virus, so one that can target human cells, and that's that jump that you talk about in the zoonotics. And then when this first started, it was very confusing because some people were just cruising through it. Oh yeah, I got COVID-19. I felt bad for a couple of days. Now I feel good. Other people were just wrecked by it. And so why can it be so symptomatic? And we're going back, a little revisionist history is now realizing that there were 2 variants, an S and an L. The S was the less common, but very mild and very unaggressive, not very virulent. And so if you had mild symptoms, you possibly had the S variant. And it's considered possible that the S variant is the hybridized one, the zoonotic variant that jumped from some other mammalian non-human host, possibly a bat, and jumped from there and was pretty mild. And then the L type is the exact opposite. That's the heavy duty, aggressive variant of it. And that may have just evolved from the S type. So again, just theory at this point. And here's a great shot. We'll do this in a little bit more detail. It's a great image. Here is the type 2. So the type 1 alveolar cells, epithelial cells that are the epithelial membrane. These are the respiratory membrane. And then there's your type 2 alveolar cell, septal cell. And then, of course, your alveolar macrophages. Those are the 3 types of cells inside the alveolus. And it's this ACE2 protein sitting on the side of this type 2 septal cell in the alveolus that is the perfect target. And the spike protein, shown in red, is adhering to that ACE2 protein and ready to burrow in, not to scale. To scale, this viral particle would be literally probably 1/1,000,000th the size of it's drawn right here in order to penetrate the septal cell. And we know that respiratory droplets are aerosolized, or put into aerosol, or put into vapor and into the air. And they're the primary means of viral transmission. And they can last on some surfaces for a decent amount of time. And so the whole thing about hand sanitizing and cleaning surfaces isn't the same as something like the norovirus, which rips through a cruise ship because it's just a touch. Any surface that you touch and then into your GI tract with the norovirus, you're instantly just consumed by it. And that's one sort of part of transmission, but this is a little different. So respiratory droplets through the air that will eventually, gravity will win, they will eventually land on horizontal surfaces, and they can last for a few hours on some surfaces, even up to a day or so, maybe even 2 days on some surfaces. And then if you happen to touch those surfaces, you're totally fine. The integument's going to protect you 100%. You can do whatever you want with those viral particles until they hit respiratory membranes. And if you happen to touch a respiratory mucosal membrane with it, then it can be transmitted from person to person. But most likely, it's just respiratory droplets that are expelled and then taken in. So in other words, somebody sneezes, and this is why the whole 6-foot social distancing. Somebody sneezes or coughs the respiratory particles into an area or droplets, you then inhale them into your respiratory tract and you are infected as such. And that's why, if the Delta variant is so contagious, it's really great at transmission that way. So what cells are bearing the ACE2 receptors that are so susceptible to the COVID-2 virus that causes COVID-19? Well, we already talked about, we'll get to the septal cells, we'll get to the type 2 alveolar, but keep in mind there's a lot of ACE2 receptors on cells lining the respiratory membrane, the upper, particularly in the nasal epithelium. Remember anosmia, COVID smell, or COVID nose, going nose blind. Fortunately, if we look at the respiratory membrane, the nasal epithelium, it was originally thought that the anosmia, or lack of smell or loss of smell, was damage to these blue cells. And these blue cells are actually, if you go all the way back to 40B, these are actually the olfactory receptors. These are neuronal tissue. These are neurons. And so that's not good. We know neurons are just tragically bad at regenerating or rehealing themselves. So that's good. The good news is they were damaging these cells that I just put an X through. The brown cells that are the supporting cells. And these are epithelial cells, so they have pretty rapid turnover. It was thought that maybe these cells are swelling, these brown cells are swelling, and maybe sort of pinching or cutting off the blue olfactory neurons and not allowing them to pick up and sense olfactory chemicals or olfactants. Good news is, with time, these supporting cells heal themselves and are replaced mitotically, and smell does return in cases of COVID. Lack of smell or loss of smell is only temporary. But really, it's just about the type 2. We saw this back in 40B. The type 1, I'll put a check mark on the type 1. That is the respiratory membrane. The type 2 are the septal cells. I'll put an arrow pointing those. And then, of course, the macrophages. But it's really these type 2 septal cells that have the ACE2 receptors. And COVID-2 is exceptionally good at binding to and infecting and penetrating inside of these type 2 alveolar septal cells, and that's not a good thing. They will be, so what will happen to the type 2 cells, if you go back to the beginning of this week, to that module on innate immunity, and we said we just throw random chemicals at pathogens invading just to try to throw stuff at them and see if something sticks. And one of those things, antimicrobial proteins we talked about, iron binding proteins, One of the ones we talked about was interferons. And so when this type 2 cell is infected, it's kind of lost. It's not long for the world. It will release interferons to help neighboring cells. Release interferons. That will cause an inflammatory response in neighboring cells. That's really great when you have an infection in the integument of your arm. It's okay when your arm blows up and gets red. That's the healing process. That's inflammation. That's helping you. That's going to be the right response. This is also the correct response, but does it make sense that the neighbor of a type 2 septal cell is a type 1 septal cell? And the last place in this universe that we want inflammation is in a type 1 alveolar cell, because we need that respiratory membrane to be incredibly thin in order to allow for O2 from oxygen from the alveoli to move through into the tissue, and then don't forget CO2 to move from the tissue, the capillary, into the alveoli for expiration and for elimination via expiration or exhalation. And if we start blowing up that type 1 alveolar cell, it's not like blowing up the dermis of your arm. That would be a good thing. And in the respiratory membrane or in the alveolus, it's not a good thing to be blowing up those type 1 alveolar cells. As once you inflame those type 1 alveolar cells, you're on the road towards two things: hypoxia and hypercapnia, or hypercarbia. You can use either term. The O2 is not going to move as easily from the alveolus into the capillary, pulmonary capillaries. You'll be left with the lower systemic or arterial PO2. That is hypoxia. That is not a good thing. The CO2 will not be able to move fluidly from the pulmonary capillaries into the alveoli. You'll start backing up CO2 in the tissue, in the alveolar, in the capillaries, in the alveolar capillaries, or the pulmonary capillaries. And do you remember what elevated CO2 does? It's hydrated into carbonic acid, and your pH will start to drop and you'll become acidotic, and that is a really bad thing. And those, my friends, are the nuts and bolts of acute respiratory distress. And when you hear somebody rushed onto a ventilator or onto oxygen, it's because they're not able to get oxygen through that thickened respiratory membrane that is being inflamed by interferons as part of the normal, natural immune response. But in this particular case, it's not a good thing. How does it get worse? Well, you remember the type 2 were damaged also before they sent out interferons. What were the type 2 doing? Remember, they were releasing surfactant out into the alveolus. And what was the surfactant doing? If I can draw in blue, I'll try to draw. Pretend this is a water molecule here. And another water molecule, not to scale, obviously. And another water molecule lining the alveolus. These all exert surface tension that are pulling inwards and trying to pull in and constrict the alveolus and make it smaller, which would be a bad thing, because less alveolar volume means less oxygen. And so the alveolar, the type 2 septal cells, inserted surfactant. I'll just kind of try to, it's a lot of the same color, but maybe you get the idea. They insert surfactant in between the water molecules, and it cuts down on the surface tension. Except that the type 2 alveolar cells are being destroyed by the COVID-2 virus. So they're not able to secrete surfactant. Therefore, surface tension starts cranking up, and your alveoli, as if the thickness of the respiratory membrane wasn't bad enough, as if this membrane now being really thick all the way out to here on these type 1 cells, as if that's not bad enough, the whole alveolus is starting to constrict inward via surface tension, and the overall volume of the alveolus is shrinking too. And that's when things just really start to get really, really bad for you. This is a great image. This is not from your text, but it's a great image again showing that AC2 protein on the receptor on the type 2 alveolar cell. And anyway, it just keeps getting worse because now you've got a damaged tissue. And there's an alveolar macrophage sitting right here. And what is the job of an alveolar macrophage? To go help damaged tissues and to try to phagocytize or eat or endositize foreign particles. And so this alveolar macrophage is not going to sit around. It's going to realize you've got damaged type 2 and type 1 alveolar cells. It's going to get to work. What do macrophages do? Oh boy. They release tons and tons of inflammatory cytokines. Histamines, bradykinin, prostaglandins, leukotrienes. Oh boy. additionally continuing to blow up the tissue, but now even worse. Some of those inflammatory cytokines will go out into the pulmonary capillary. What are they going to do? They're going to in this case, sorry, not broncho, they're going to vasodilate. They're going to increase those capillaries. You're going to get more blood flowing through the pulmonary capillaries. Not necessarily a bad thing, but we know what they're also going to do. What are those inflammatory cytokines going to do? We saw this in previous modules. They're going to open up the intracellular clefts, and they're going to increase the permeability of the pulmonary capillaries. That's going to mean more fluid out into the surrounding tissue around the alveolar. Hello, pulmonary edema, swelling, all this fluid meant to be in red, all this excess fluid, as if it weren't hard enough with the respiratory membrane in general, all this excess edema, this excess fluid causing edema is just going to make it worse and worse and worse. But wait, we're not done, because now the neutrophils cruising through the pulmonary capillaries, they're sensing edema, they're sensing infection, they're sensing cytokines, they're moving positively chemotactically towards the cytokines. They're going to enter the alveolus and they're going to do what they do, which is just shoot first and ask questions later. Remember, neutrophils first on site, most effective early on, incredibly non-specific, just bombing away. And it's great, neutrophils and macrophages bombing away chemicals and also doing phagocytosis, which is great. But what sort of chemicals? Those neutrophils are going to go into the alveolus and just bomb ROS, reactive oxygen species, superoxide anion, hypochlorite ion, the most toxic substance in the human body, arguably. And it's just going to rip apart your tissue, rip apart your alveolus, and it's just going from bad to worse. And again, these are the nuts and bolts of acute respiratory distress syndrome. So what are our treatment options? Antivirals in general. Remdesivir is a generic. It's not really specific to COVID-2. Antivirals in general work to just sort of knock down viruses and to try to block transmission. They're nasty. Antivirals are nasty. You take an antiviral, you're going down hard because it wipes out a lot. Some people refer to antivirals as anti-life. They just kill so much, but it is a treatment. Dexamethasone is an incredibly potent steroid anti-inflammatory. We talked about the problem being that respiratory membrane is inflaming. So an anti-inflammatory like dexamethasone will be really helpful in getting your respiratory membrane back down to the correct size and cutting into that severe acute respiratory syndrome effects. Decrease the size of the respiratory membrane. Try to get more O2 and CO2 flow in the correct directions. Try to bring up low O2. Try to correct hypoxia. Try to bring down hypercapnia. Try to bring back up blood pH. How do you gain artificially acquired active immunity? You get a vaccination. And so a lesser version, a truncated, a blunted, a neutralized, a non-virulent version, a genetically modified version of that virus is introduced so that your body can see the spike protein and it can make antibodies against it. It can see the opponent without it being at full speed or even close to full speed. You get a reduced and injured opponent so you can really load up on all the correct cytotoxic T cells, all the correct helper T cells. You can clonally select all the great B or plasma cells that you need, plus the memory cells, which is going to allow you to deal with a second or a third infection. So technically, 2 shots, the vaccine and the booster should be enough. A third booster, if we're talking about possibly immunocompromised or immunosuppressed people, might be in the cards too. And then how do you get artificially acquired passive immunity? Well, monoclonal antibody treatment is something that you'll start to hear a lot about. You take plasma from a person who is recovered, who is very generous in donating plasma. Why it's so important. A recovered COVID-19 patient who donates plasma is a miracle worker, because from there you can isolate the exact antibodies that are necessary, spin them down, isolate them, and put them into another vulnerable patient, particularly someone who is immunocompromised, immunosuppressed. Really, in most medical communities and all medical communities, it's beyond debate that the vaccination is the more effective and it's cheaper, but monoclonal antibody could be a complement treatment for somebody who is a bit immunosuppressed or immunocompromised. So, anyway, kind of what you need to know about COVID-19, and thanks for sticking with this and watching it. We'll jump right to the standard clinical applications of the lymphatic system and immune function.